thanking everybody who's logging into the workshop now. This started off as an event in 2020, a 5-day event on open source software. The workshops proved incredibly popular, and so we decided that we would continue this throughout 2021, and we've had a monthly workshop on various aspects of open source, either software tools or online resources, etc. They've all been extremely popular, and we're very much aware that people are now using these as an invaluable resource. We're very fortunate, I think, and very timely, I suppose, in that this month's webinar is going to be on the Protein Data Bank. As you can see, we're celebrating 50 years of this absolutely fabulous resource. I think one of the things that anybody who's involved in structure-based drug discovery or any type of structural biology, you very quickly become aware that this is probably the premier resource for this sort of information. And so we're absolutely delighted that we are going to get this sort of discussion and also workshops on this invaluable resource. So no more from me. I'd like to hand over to Stephen Burley, who will be your host for the workshop.
Thank you very much. And we at the RCSB Protein Data Bank and Protein Data Bank Europe are extremely grateful to the Royal Society of Chemistry. Let me just share my screen, and we can get moving. So my name is Stephen Burley. I have the pleasure of and honor of serving as the director of the RCSB Protein Data Bank, which is the US data center part of the Worldwide Protein Data Bank Partnership. We're based at Rutgers University and at the University of California, San Diego, and at the University of California, San Francisco. And today with two of my colleagues, Ezra Peisach and Chenghua Shao, we will be presenting the first session, first of two sessions. The second session will be led by Samir Volankar, who is the head of Protein Data Bank Europe, the European Data Center for the Worldwide Protein Data Bank, and he and his colleagues will be presenting on Thursday at the same time. So my job is to set the scene and explain how we got to where we are today. And the story begins before any of us were born, I think, in 1935, when Dorothy Crowfoot Hodgkin, subsequently a Nobel laureate in chemistry, recorded the first X-ray diffraction pattern from a protein crystal. And that single experiment established that proteins have defined structure. She was a student of J.D. Bernal, the sage of science, who played a central role in establishing the discipline that we now know to be protein crystallography. Two other pioneers are important in this early stage. Sir John Kendrew and his team determined the X-ray crystal structure of myoglobin. That was the first protein to be visualized at atomic resolution. And he used many of the methods that were developed by his colleague Max Perutz, who was working on the hemoglobin crystal structure. And you can see on the bottom right of the slide, you can see Perutz holding the hemoglobin alpha-2 beta-2 heterotetramer, and Kendrew holding a somewhat larger scale rendition of myoglobin monomer. Both of them, of course, are oxygen binding proteins. Myoglobin is for storage, hemoglobin is for carriage. The first enzyme structure to be published was that of lysozyme, depicted here in a painting that was from the work of Irving Geis, who became the premier molecular illustrator in structural biology for many years. And then other enzyme structures that followed soon thereafter included ribonuclease, depicted here, and from Fred Richards' lab, and carboxypeptidase A from the Lipscomb lab at Harvard. Everyone who is working in protein crystallography today is standing on the shoulders of these giants: Kendrew, Perutz, Phillips, Wyckoff, Richards, and Lipscomb, making giving further testament to the truth of Sir Isaac Newton's statement: "If I've seen further than others, it is by standing upon the shoulders of giants." Among the data pioneers we need to recognize, thinking about the origins of the Protein Data Bank, are the individuals who were involved in establishing the Cambridge Crystallographic Data Center, which I suspect many of you are using. And depicted here is in the center in this left-hand photograph is Olga Kennard, David Hartley, and flanked by David Hartley and Frank Allen. This is the building in which, the house, in fact, in which the Cambridge Crystallographic Data Center was established, and this is its current home in the middle of Cambridge in the UK. I suspect many of you use the Cambridge Structural Database, which is the small molecule counterpart of the Protein Data Bank. The 1960s saw in the world of structural biology and macromolecular crystallography a push among open access data activists to begin to promote the sharing of X-ray crystal structure data for proteins. And some key movers in this early open access data movement included Edgar Meyer, Gerson Cohen, and Helen Berman, who went on, as you'll hear, to lead the RCSB Protein Data Bank here in the US. They had many meetings and exchanges through what we now refer to as snail mail. The American Crystallographic Association took a supportive role, and here's a page of signatures that were generated at the time that people were thinking about establishing a PDB. This is 1971 when this was happening, the 1960s culminating in the 1971 foundation of the PDB, well before the term open access was being used, and certainly long before things like the fair and fact principles came into the modern vocabulary. So the PDB, as I said, was established as a global enterprise in 1971. Its place of birth is nominally the Cold Spring Harbor Laboratory on Long Island in New York State. And there was a meeting there in early June of 1971, structure and function of proteins at the three-dimensional level. It was organized by Jim Watson, of the Watson and Crick fame. And co-organizers included Aaron Klug, who went on to be a major player at the LMB MRC, Bill Lipscomb at Harvard, Max Perutz also at LMB MRC, David Phillips at Oxford, and Fred Richards at Yale. And here are a number of photographs that were taken at the Cold Spring Harbor Symposium. If you have not been to a Cold Spring Harbor Symposium and you have an opportunity to attend, I strongly urge you to do so. The grounds are really quite spectacular in the setting on Long Island Sound. Here is Dorothy Hodgkin depicted in one of these photographs. So that push at the meeting led to a decision by Walter Hamilton, who was actually, sorry, I should have who is here in this photograph. And Walter Hamilton was, took the initiative and actually established the Protein Data Bank at Brookhaven National Laboratory on Long Island, just a little bit further east of where those pictures were taken. There was an announcement on October the 20th that was published in Nature New Biology, just a single column. It described a collaboration with Olga Kennard at the Cambridge Crystallographic Data Center, and Edgar Meyer, one of the activists that I mentioned earlier, was responsible for creating the original technology stack that supported the sharing of approximately 7 X-ray crystal structures of proteins that individual labs were willing to share at the time. We now turn to the evolution of the PDB data centers worldwide and the basis of the modern protein data bank. So initially, the PDB was at Brookhaven. It was frequently referred to as the Brookhaven PDB from 1971 to 1978. And you can see that the team grew and the mode of photography changed from black and white to color. In 1999, the National Science Foundation, which was the principal funder of the Protein Data Bank in the US at the time, made the decision to move the PDB from Brookhaven to Rutgers University. This was the what became the Research Collaboratory for Structural Bioinformatics, RCSB Protein Data Bank, which is the US-funded PDB data center of the Worldwide Protein Data Bank today. And here are a number of photos showing the evolution of the team. This is the last pre-pandemic photograph that I could lay my hands on. The original performance sites for the RCSB PDB were at Rutgers, at the San Diego Supercomputer Center, at University of California, San Diego, and at NIST, the National Institute of Standards and Technology. NIST subsequently dropped out of the PDB team and was, and recently the University of California, San Francisco joined. In 2003, the RCSB Protein Data Bank and data centers for the PDB in Japan and Europe, so Protein Data Bank Japan and Protein Data Bank in Europe, decided to actually establish the Worldwide Protein Data Bank Partnership. This consortium of 3 has grown to 5, now including the Biological Magnetic Resonance Bank, BMRB, and the Electron Microscopy Data Bank, EMDB. Working together, these 5 data centers worldwide ensure that the data are freely and globally available. We collaborate on data deposition, data validation, and annotation, and we jointly manage, these 5 organizations jointly manage 3 core archives: the Protein Data Bank that we're talking about today, the Biomag, ResBank, BMRB, and the Electron, which includes experimental data for nuclear magnetic resonance of biological macromolecules, and the Electron Microscopy Data Bank, EMDP, which includes the electric Coulomb potential maps for 3D EM structures. Each data center provides its own website, and these websites offer different services and different views of the data. And you'll get a sense of that over the next 2 sessions as you see screenshots coming from the RCSB Protein Data Bank and Protein Data Bank in Europe, our collaborators in this event with the Royal Society of Chemistry. Important in the history of the PDB was the evolution of both policies and data standards. In the late 1980s, there was a move within the structural biology community, Macromolecule, to ensure that data were for biological, structural biology data for biomolecules was going to be faithfully captured. And the discussions revolving around data standards involve the International Union of Crystallography, its American Crystallographic Association member, and a committee that was established by Fred Richards. They contacted the editors of scientific journals and the funding agencies, and the goal was to mandate structure deposition at the time of publication. And this was something that was very unusual at the time and set the stage for what we know as the open access data ecosystem in biology. There were articles in Science magazine, letters were written, and eventually the funders in the US took the decision based on the input from the community that they should require US-funded structural biologists to contribute their data to the PDB. at the time of publication. And this became and continues to be a condition for funding in the United States, and it became and continues to be a condition for publication in major scientific journals such as Science, Cell, Nature, etc. The other important aspect of the history of the PDB concerns community data standards, and you'll hear a little bit more about this from Ezra Peisach. The macromolecular crystallographic information file, mmCIF, is the basis for all of the storage and archiving of information in the PDB. The first draft of the mmCIF standard appeared in the mid-90s. And in 2011, the PDB Exchange macromolecular crystallography information format, PDBx mmCIF, was adopted as the master format for the PDB, providing definitions not only for X-ray, but for NMR and 3D EM. And as you'll hear from Ezra, for other types of experimental structural science and de novo protein structure prediction. The use of PDBx mmCIF files is now mandatory for data deposition to the PDB, and this has greatly improved the speed with which we can process the incoming data and the quality of the data sets that are being contributed by more than 50,000 depositors around the world. Another feature of community engagement by the Worldwide Protein Data Bank has been data validation and ensuring that community standards for data validation and structure validation are enforced through the activities of a series of task forces on X-ray, nuclear magnetic resonance spectroscopy, 3D electron microscopy, and more recently on small angle X-ray and neutron scattering, and so-called integrative or hybrid methods, wherein no one technique is sufficient for determining the structure, and multiple data from multiple experimental methods need to be brought together in order to be able to determine a 3D structure. And some of you may recognize the faces in these photos, reflecting meetings of the various task forces. The science has evolved enormously since the 1970s. PDB contained structures of hemoglobin, myoglobin, small enzymes, RNA. In the 80s, we saw structures of deoxyribonucleic acid and viruses. In the 1990s, we saw complexes of proteins recognizing DNA. And then in the 2000s, there was a huge surge of new information concerning macromolecular machines, starting with the ribosome and continuing to today. This evolution in science was accompanied by an evolution in technology for structural biology. We started out with very crude measuring devices and are now using state-of-the-art robotics to support structural biology sample preparation and structural biology experiments. The community has also grown enormously. There were probably a few hundred macromolecular crystallographers worldwide operating when the PDB was established at Brookhaven National Lab in 1971. Today, we have records of more than 50,000 depositors of PDB data to the archive, and the community has been involved at every step of the evolution of the PDB and the Worldwide Protein Data Bank partnership, which supports, as I said earlier, the PDB archive, the Biomag ResBank archive, and the Electron Microscopy Data Bank archive. So the PDB today is the single global archive for protein, DNA, and RNA experimental structures. As of this morning, we in the Worldwide Protein Data Bank, that means RCSB PDB in the US, Protein Data Bank Europe, Protein Data Bank Japan, Electron Microscopy Data Bank, and Biomag ResBank are supporting open access to nearly 184,000 structures, having grown from just 7 X-ray structures at the beginning of the PDB back in 1971. It's not an exaggeration to say that the Protein Data Bank was walking the walk when it came to things like the fair, fact, and trust principles. And we've actually been accredited, the Worldwide Protein Data Bank has been accredited by the Core Trust Seal, non-governmental organization that adjudicates compliance with these principles. The PDB was founded in 1971. The first paper describing the FAIR principles was published in 2016. It was a long time coming, but you can see in the open access data ecosystem that supports all of biological and biomedical research today, the impact of the PDB on the inception of this important resource for the community that's today used by many, many millions of people worldwide. The users of the PDB come from every sovereign country recognized by the United Nations, and structure depositions to the PDB come from every inhabited continent on the planet, the sole exception, of course, being Antarctica, where there's no active X-ray, no active protein crystallography or electron microscopy or NMR spectroscopy going on. Our program today will feature Ezra Peisach and Chenghua Shao from the RCSB Protein Data Bank. Ezra will speak briefly about the data standard, the PDBx and MSF data standard, and PDB data deposition using the global OneDep system that was developed by the Worldwide Protein Data Bank Partnership. And then Chenghua Shao will go on to talk about small molecules in the PDB. This should take, including my talk, no more than a total of an hour. We'll have a short break at that point to give everybody a chance to stretch the legs. And then Ezra and Chenghua will conduct a set of tutorials on the use of the OneDep system for deposition. And for this particular audience, we've included a tutorial on small molecule information that's stored in the Protein Data Bank. These are the small molecules that are found bound to protein and nucleic acid structures. On the 18th, on Thursday, same bat time, same bat channel, the Protein Data Bank in Europe will take the lead, and you'll hear from John Beresford, Jack Turner, James Tolchard, and the director Samir Valankar. And then at the end of the session on Thursday, there'll be a brief roundtable discussion involving Samir and myself talking about the future of the PDB. So with that, I'm going to stop. I'm going to thank you for your attention. I don't know if there are any questions in the chat, but seeing none, I'm going to move directly to Ezra Peisach. He's a protein crystallographer by training and a longtime member of the RCSB Protein Data Bank. Ezra, the floor is yours. You need to come off mute, Ezra. Yep.
I know, I just realized that. Okay. So thank you, Stephen. I'm going to talk briefly about the PBX and MSF data standard, and then we'll talk briefly about data deposition within the OneDep system. So the first, to repeat what Stephen said, among our partners within the WWPDB, we are all working within PBX and MSF. We all share in the responsibilities for processing data, and we all share the same data on the output. We may augment it with our websites, but the formal core data, which you could download from an FTP site, is the same. So I know some of you are from the small molecule world, so I'm just going, this may be very much a review for others, but I'm just going to make sure that we're all talking about the same language. So when we're talking about a PDB, a protein data bank macromolecule, we are talking about a set of atoms which are each represented by what we call a residue, which could be, say, an alanine, a sulfate ion, sulfate ligand, It could be a metallo ion. Residues are combined together in polymers to form chains, which we are labeling them with a unique identifier. And then these chains of same sequence are represented by an entity. An entity has the same sequence, the same molecular source, same reference sequence, and other metadata like molecular weight among all of them. So the PDB format, which has been in wide use since 1976, is the fixed width 72 character wide data format. This is a sample of part of a PDB entry, 3LTQ. And I just want to point out a few features of it. I'll go down to the atomic coordinates section down here first. This is the part which many people use. We have a series of atom names associated with a residue, with an XYZ coordinate, a sequence number, occupancy, and temperature factor. We have a series of metadata within this PDB file format, where this is showing some links between a metal and various amino acids. And we have, for instance, unit cell. There's a lot of other metadata here. One of the challenges that you could see is that everything is in a fixed column. Sometimes we've had users complain for very large structures, where all of a sudden these columns combine together. For instance, if this was 3.410 and this is minus 149, there would be no spaces. And they say that the file's broken. They don't understand the concept of fixed columns and fixed column width. The metadata have all been agreed to, and anytime there ever has to be an expansion of the data format, then there's an issue. It used to be PDB format was updated infrequently as different references had to be updated, and now we've pretty much frozen the PDB format. So some of the other limitations of the PDB format. Well, there's only one column for the chain ID. And there are only 5 columns available for serial numbers. So this means you can have only essentially just under 100,000 atoms and 62 chains, polymer chains, because you have lowercase a through z, capital A through z, and then the numbers 0 through 9. And you could say, oh, this is no big deal. We compass everything, until you wind up with something like a ribosome. Ribosomes are macromolecules that take the messenger RNA and tRNAs and put together polymer chains within the cell. And these tend to be pretty large. These ribosomes really expose limitation in the PDB format because we could not represent them properly. So we had a little bit in the early 2008 or so, we came up with the approach of splitting these into multiple PDB formatted files with multiple PDB identifiers and some instructions on how to put them all back together again. But this meant that you could not validate all the data together. You couldn't look at the whole picture at one time without loading multiple files. So in the late 90s, from around 91, 97, there was the development of PDBx and mmCIF. So for some of you who are familiar with small molecule crystallography, the original CIF, crystallographic information file, which was developed or approved in 1991, and it is still used in small molecule crystallography today. So it's perfectly good format. The goal was to expand it to macromolecular CIF, a format that included an extended set of data items to make it compatible with macromolecules. We also added the term PDBx. So this is more flexible than a PDB format. Here's a sample on the right of what a file might look like. We have a category with data items. We could expand these data items as the science evolves if we need to have different items added in. We are able to accommodate any width column. So we are not limited by the field width of the coordinate atom or even the width of a polymer chain label. We could also readily expand this with method development. In the last few years, serial crystallography has become much more popular, and we are now collecting new metadata for that. That could not be incorporated into the legacy PDB file format because now that would be breaking the format that everyone is relying upon, the fixed standard. But with mmCIF, we could easily add these data items in a unique way, and people who are interested in them could then mine them. This file is readily parsed, searched, and edited on a computer. Right now, I think there are about a half dozen or more parsers that could parse this file, including some popular Python, Java packages, as well as C++ and other computer languages. This just gives you an example of how big this would be. This was partially what motivated us to move in this direction. This is the entry 3J3Q, which is the capsid assembly of HIV1. If we were to go back to the old split file into multiple pieces, it would take about 25 files, which was very unsatisfactory for everyone who might want to use it. As Stephen mentioned, we care about data standards, and we established various working groups to help ensure that we weren't going off on a limb and making sure that people in the field could give us good guidance. So the PBX mmCIF working group was brought together where we had experts and method developers who knew the field, who were experts. Right now, we still are meeting, and they're experts from all aspects of crystallography. Oops, sorry. We've also established an NMR Task Force working group. They gave us advice in terms of how to come together with an NMR exchange format called NEF, which is similar to NMR star, but has different data items. There was an expansion to support the small angle scattering database with something called CICAG, which is a similar type of dictionary, but it has key items which are important for the exchange of the small angle scattering data. And if you go to their website, you could download data in that format. We also support a number of dictionary extensions. So if you think about it, PBX mmCIF is sort of like a basis, but as different fields need different things, they expanded the dictionary in their own manner. So if you're thinking about anything with coordinates XYZ, PBX mmCIF is great, but then individual archives extend them. So there was the model archive extension, which has been used at the modelarchive.org, which describes information about molecular modeling. The modelarchive.org contains well-loved, curated methods defined by just computational means, maybe using templates from PDB structures. But it's now been used more commonly in the news, AlphaFold, which came out in Nature from Europe in the last few months. There are other ones that are using Rosetta Fold and ModBase. These are all complete computational methods, but they're relying on the base PDBx mmCIF to form the foundation, and then this extension dictionary, model archive extension, to give them the extra metadata that they need to describe what they've done. In the more experimental world, we have the integrated hybrid methods, IHM, which is available on our PDB dev website. This is a combination of in silico work plus biophysical data, such as infrared or cross-linking experiments, or it might be bringing together structures from different, it may go totally in silico from folding up a model, or may take a model from the PDB and combine it with the extra information to construct bigger constructs, like as in the spliceosome over here using cross-linked and mass spec data. This has its own supported dictionary extension, which layers on top of the PDBx mmCIF. We also maintain a website, mmcif.www.pdb.org, and this is sort of what I call one-stop shopping for information about PDBx mmCIF. We have information for learning more about it, including some of the early history about it, and some references and list of software resources. The software resource includes CIF parsers, visualization tools supporting mmCIF, and including some of the tools that we have developed at WWPDB. So you may think that we're only thinking about macromolecules, and that's not totally true, because a macromolecule, as I said, a residue, is really a series of chemical components put together in a polymer sequence, similar to a charm bracelet where you have a bracelet with different charms along the length, and then various other what we'll call ligands floating around in the milieu in the crystal. We have established the chemical component dictionary, CCD, which is available at our FTP site. And this contains definitions of what every chemical component that we reference it within the archive. And you can see it's based on the IUCR core CIF definitions partially, but it's based a lot on having identifiers, names, dates, formulas, formula weights somewhere else down here, not shown. We describe chemical bonds within the substructure, and we have descriptors, including SMILE strings. So, and this is a searchable resource available from the archive or from Ligand Expo. Also, we have an archive of what we call Biologically Interesting Molecule Reference Dictionary, BIRD, which is a series of annotations that link together polymers which are antibiotics or ligands, or inhibitors. Some of these are very unusual, but you should have a way in order to search for these larger constructs. Our structure factor files for X-ray, neutron, electron crystallography, and powder diffraction all adhere to the same dictionary standard format. So it's not a binary format. You don't have to guess which refinement software was being used because everything has been standardized with the same type of key and value to describe what's in each data table. The PDB established a series of validation reports. This will be discussed in much more detail on Thursday, but we provide the information in PDF, but in order to be machine readable, there's also an XML and an mmCIF type dictionary that supports it. So you could use that to mine data as well. So just want to sort of give a comparison between the IUC Core CIF versus the PDBx mmCIF. They're very similar. And in fact, most of the IUC Core is incorporated into the PDBx mmCIF. So you have things that describe features which are not necessarily in use by our archive, but you could use it. For instance, orientation having to deal with diffractometers and orientation of crystals. We could support that if we wanted to because it's in there. The names are similar. If you looked at a CIF from the IUC Core, you'd see something like atom site, Cartesian X. So it's a category within a category. So the category might be an atom site, and then it's represented as star. Then there's Cartesian X, Cartesian Y, versus the mmCIF one is more of formally a category and attributes. Both standards dictionaries have the concept of parent-child relationships to tie data together. So in PDBx mmCIF, we make extensive use of this to ensure that we have the right links between categories. For instance, if you have within your atomic sites, I showed you how to residue. Well, that's a chemical component. Well, then your model file better have information about that chemical component somewhere in there. So then they link together. The dictionary also makes extensive use of mandatory keys. It's very explicit within the dictionary. And if you ever want to load these tables into a database, these mandatory keys could be used for indexing. Sorry. Where are we going with this? The old PDB legacy format? Well, you should be aware of a few things. As I've hinted before, there are limitations in the format due to the fixed column width within the file. Well, there's some other things that are coming up in the next few years that you should be aware of. In the next few years, we'll start issuing 4-letter character codes for the chemical components, residues, or ligands. Once that happens, a PDB file cannot be produced because there's only room for 3 characters. We'll also be eventually expanding the PDB IDs to a prefix PDB and then 8 characters format. Once we run out of PDB IDs that we are issuing right now, which is only 4 characters, that will mean that we cannot represent this in the PDB file format, at which point PDB format files will not be able to be produced for these newer entries. Thereafter, newly deposited PDB entries will only be available in PDBx mmCIF format. We have worked with various software developers to be ready in this position. We are continuing to work with them. So in summary of this section, the PDBx mmCIF dictionary is very similar to the IUC Core. it's essentially just an extension of the original IUC Core definitions to include more of a macromolecule polymer type environment, and how describing more formally where the material is coming from, source organism, etc., and that the legacy PDB format will eventually become obsolete. I'll pause for a moment before moving on to the next section. If I don't see any questions this moment. So if you have any questions, I'll stop at the end of the next section and we'll talk through the Q&A tab. My understanding is you could type in a question there.
Yep, the Q&A tab is empty at present. Ezra, please proceed.
Right. Yeah, I'm just reminding people that they could talk there, ask questions there. So moving along. So I'm going to talk about our data deposition system called OneDep. This was a mutually funded and developed project among our partners to unify the deposition of data in the same mechanism. Prior to this development, there were similar but different deposition systems depending on which site you went to. The biocuration practices were very similar but not identical. This helped to ensure a uniform mechanism among all our partners. So for crystallographic structures, electron, X-ray, and neutron diffraction, you are now required to deposit PBX and MSF, as we described the format. We have tools that help people convert to it. We also worked with the software developers to ensure that the common refinement packages all support doing this directly now. And if we have another one, some of the more rare ones, we have tools to help convert it over to that. Within the OneDep deposition and biocuration system, we use PBX and MSF exclusively to do all our processing. And we are returning atomic coordinate files back to the depositor in mmCIF, along with a PDB file if possible, for them to review it. So the PBX and MSF framework was used. There are several steps for a deposition. It's like preparing your taxes. You get all your stuff together before you start. You could use what we have our standalone validation servers to check your data, look for any obvious problems before you even start your deposition. These are anonymous servers that are available globally for people to use and check their work before they start a deposition. They'll deposit their data. We will annotate, biocurate the data, and we will send it back to you within the same deposition system through a communication channel. Eventually, when it goes out to the public, it's put into into our FTP archive, which is shared among our partners. The workload for processing is shared globally among our partners right now, based on the geographic region of where you are submitting your data from. And the processing that we do is all standardized and our procedures and policies are made available publicly online at the www.pdb.org website. I'll get back to validation in a little bit. The OneDep deposition system looks a little like this. When you come to our website, you are going to be telling us which country you're coming from. You will then have the ability to upload your files. We'll talk about this in the tutorial later. Upload your files, and there'll be some basic checks, sanity checks among your experimental data and your model, including, for instance, let's think very simplistically. Does the unit cell between your model and your structure factor reasonably agree, or the space group reasonably agree? If they don't, then there's probably something wrong. We will then provide you with a preliminary validation report. You get to review it, and then you get to submit the data. When you submit the data, you'll get your PDB ID and DOI or other accession codes given to you right away. This is unlike when I first started, where you had to wait 3 or 4 days to get your PDB ID. So there's no reason not to get this done before you submit your paper. In 2020, we had about over 15,000 depositions, and this was increased over 2019. And as people, I think, were locked up and staying home, they had a chance to actually work on their papers and finish up any refinement they had to do, as the labs were closed for active work for a while. So as I mentioned and hinted, The workload is balanced geographically across WWPB sites, roughly 50% to RCSB in the United States, 30% to PBE in England or UK, and 20% in Japan and Osaka, Japan. We use a modularized workflow where we go through a series of fixed steps, including working on data, ligand processing, where we look at your ligands that you might be giving us information, adding them to our CCD. We work through sequence. We check your sequences against representative sequences in the UniProt or NCBI nucleotide databases. We identify possible mutations or conflicts, which may be real, they may be errors. There are errors in the sequence databases which come out eventually. We provide some extra value-added annotation, including secondary structure, links, and others. We will provide a validation report for you, and then we communicate it back to you. During this process, we'll produce what's called a confidential validation report. This validation report is what we want people to submit to the journals. This means that we've taken into account your ligand definition, and everything has been knit together, so to speak, in your final presentation. It would be like you're about to release it. So I kept talking about validation. So what we did was we adopted recommendations from a validation task forces and workshops. We have them on X-ray, NMR, EM, ligand validation. The software was developed with that in mind. Data are presented as either a PDF file, XML, and mmCIF, PBX and MSF formats. Validation reports are offered at 4 different stages. I told you could do the anonymous standalone validation server at validate.www.pba.org. There's also a standalone API, anonymous API service that you could submit them fully electronically. And then you'll be given different validation reports, depending on what stage of your deposition they are. So you can do it before you start your deposition. You get during your deposition, after biocuration, and the public release. Each have different watermarks and signifying where they should go. But I want to emphasize it's after biocuration. The confidential validation report is a report that many journals are now requiring at the time of your submission. So some key features of the validation reports. Again, more details will be on Thursday. We have a number of sliders which give you an overall quality, including polymer residue geometry, chirality, close contact geometry, ligand geometry. We will have more details about per residue plots. In this case, it's an electron microscopy structure, and you can see there are individual residues or missing residues in the sequence. You just don't observe them, it happens. You'll have residues identified that may have some issues. And for X-ray, we'll give you a quality fit of how well the data fits to the model with looking at electron density maps. Over the last few years, there have been some improvements in partnership with global phasing. For ligands of interest, we will highlight them and give you a 2D representation and 3D representation in electron density maps. These plots would tell you if there seem to be any strange torsion angles, rings that aren't flat, bond angles, or bond lengths. Using the Cambridge small molecule database, we use a program called Mogul to analyze fragments and try to say, do we have the right geometry for these unknown compounds? So here gives an example of 2 NADP ligands. The first one is better. You can see that there's very little outliers in terms of geometry, and you can see it fits very well into electron density maps. On the other hand, you have one which looks pretty good geometry-wise, but then you look at the electron density map and you see it's sort of ropey. This is something that a reviewer could then look at and maybe give feedback back to the depositor who's working on submitting their paper. More recently, we've added support for branch change carbohydrates. This is in partnership with Noguchi Institute. Glycosylated proteins have a critical role in some of the functionality and in terms of affecting geometry and accessibility. So we have formalized a representation which is standard within the glycobiology field to describe a branch chain, not nonlinear polymer, representation of this happening on protein chains, and we are providing the same type of analysis. We are expanding more and more into 3D EM and NMR structures. In the last couple of years, we've added 3D EM where you can see a map and model, electron density map and how well the model fits, and descriptions of, at the particular contour level, how much of your data are included with NMR. As we get more structure, formalized restraint information that's standardized in NEF or NMR star formats, we can now start doing real restraint analysis, which has been left out for the most part because each software package had their own format. So moving forward, the OneDep system will continue to improving on the validation. We'll continue to evolve based on emerging experimental methods. So crystallography is still growing, and we're getting more data from them and in terms of what their needs are. 3D EM, there are going to be changes to support more metadata, more controlled vocabularies, and we will eventually be integrating more with the integrated hybrid methods. And that is all I have to say, and I'll answer any questions right now. I see Alex Henderson. Do we have any interaction with the Helm hierarchical editing language for macromolecules for the representation? I don't believe so. I'm not familiar with it. Does anyone else on the panel happen to know?
I don't know, Ezra. Sorry. You'll hear, Alex. You'll hear in a moment. We will we'll check with our colleagues and get back to you. You will hear from Chenghua Shao now, who will talk about small molecules in the PDB, and you'll see that both Smiles and Inchi are supported by the small molecule data management system that's part of the protein data bank. So let's move on, and we'll hear from Chenghua Shao, who's also trained as a protein crystallographer, and he has during his time at Rutgers pursued a master's degree in statistics and has emerged as our expert in matters pertaining to validation, and his expertise in chemistry has allowed us to make some advances in terms of understanding quality of small molecule structures in the PDB. So you'll hear from Chenghua now.
Yep. I'm going to share my screen. Can you see my screen? Okay. Thank you, Stephen and Ezra. My name is Chenghua Shao, and I'm working at RCSB Protein Data Bank as for data curation and data analysis. Before coming to PDB, I was a structural biologist, and I saw structures of protein ligand complex, such as annexin with heparin glycan and enzyme with their substrate. So today I'm going to talk about small molecules in the PDB. We'll start with the introduction to the holdings of the small molecules in the PDB, and then I will talk about how we annotate the small molecules to ensure data accuracy and reusability. And finally, I'll talk about how to access and analyze the chemical data in the PDB. And shortly after this talk, we'll have a tutorial on that as well. So, I'm sorry, I got wrong click. Just start from here. So from Stephen, you have heard the long history of PB as a data archive of macromolecule structures. It's also important to understand the interaction between the proteins and small molecule ligands. Actually, from the very beginning of the PDB, small molecules were part of the macromolecular complex structures For example, on the right side, you see two examples of coenzyme NAD and iron sulfur clusters in the structures deposited in 1975. Now there are over 180,000 PDB structures. Among them, over 70% contain non-polymer small molecules. And they represent about 35,000 unique chemical components, with each described in the PDB chemical component dictionary. There are drugs, for example, as you can see from the right side, the penicillin G and remdesivir, that's the COVID drug, which is a structure resolved and deposited during the pandemic. And there are also cofactors, activators, inhibitors, etc. You may notice that the number of unique chemical components is much smaller than the number of PDB structures. That's because one ligand may be present in multiple PDB structures of either the similar or different targets or under different structural conformation. From the periodical table, I have circled the ones that is present in PDB. As you can see, about 70% of the elements are present in the PDB. And if you only count the lighter elements below 83, it's more than 90%. On the left is the frequency of each element in their presence in the CCD, PDB CCD component dictionary. And you can see that organic elements such as hydrogen, carbon are abundant, of course, but you also see things like halogen. They are also frequent in the PDB as either as a replacement of the derivative, replacement group of derivatives. And going down, you see things like myols, usually work either as ions or cofactors. The PDB chemical component dictionary contains rich data content. And I will list some examples here. Each unique component molecule is identified with 1, 2, 3 alphanumeric letter code, and together with the name and synonyms. Each definition contains the list of atoms, with each atom uniquely labeled together with their bond. We also include information such as the model coordinates from the representing PDB entry and SMELT and inches descriptors, which I will show in an example in the next slide. In terms of data format, as Ezra said, the CCD data is primarily annotated in the PDBx and mmCIF format, but we also provide other format distributions such as PDBml and SDF. I want to stress that there's only one CCD that is maintained by all the PDB partners. And this CCD is different from the Cambridge structure database that is maintained by CCDC. Now here's the example of the CCD definition of penicillin G in mmCIF format. You can see first the unique identifier PNN, the short letter code for this ligand, and the formula and formula weight are recorded, and other things I skipped. Going down, you see the atom list. Each atom is uniquely labeled with their model coordinates and idealized coordinates. And there's a bond order all explicit, and the chemical descriptor of SMILES, canonical SMILES, INCHI key. Now this chart shows the growth of the CCD, with the dark blue for the yearly growth and overall head for the accumulated numbers. And what you see is it has grown rapidly, especially in recent years, which each year having more than 2,000 small molecules new to the PDB archive deposited. And we have now more than 35,000. In 2020, there are actually 400 small molecule fragments deposited together with SARS-CoV-2 proteins in searching of the COVID drug. In terms of molecular weight, What you can see in this distribution chart is most of the CCD ligands are between 300 and 400,000. Actually, more than 90% of the CCD ligand are between 100 and 700,000. I'll give you some examples here. For example, phosphate ion is about 90,000 in the first bar, glucose 180,000 in the second bar, and penicillin G is 330,000, and ATP is about 500,000. The mean and median of the distribution was 370 and 350,000. Now, there are also different types of molecules in the PDB CGD dictionary. There are about 1,400 amino acids, for example. With the example shown on the right side, the phosphotyrosine under the CCD ID PDR. And there are 700 nucleotides and their derivatives. For example, the 2-fluoroadenosine monophosphate. And you can see the halogen here is the replacement group. And there are also about 1,000 monosaccharides. The example here is N-acetyl beta-D glucosamine, which happens to be the most popular monosaccharide in the PDB because they are the essential part of glycan. And there are about 200 common ions, etc. Although the vast majority of the CCD ligands are non-polymer components that associated with macromolecule, such as the TTT here, that is SARS papain-like protease inhibitor. But we also include the amino acids or nucleotide that can be present either as a residue in the sequence or as a standalone free-floating ligand. Now, I indicated that CSD is different from CCD, but there are overlaps. About 8% of CCD, that's 2,700 ligands, are shared between the CCD and CSD. And about 17% of CCD, that's 6,000 shared between the CCD and drug bank, which confirms that the interaction between the drug and their target is one of the focuses of structural biology study. And about 3% of 1,000 ligands shared among all three databases. So in addition to the CCD, WPD also provides a complementary chemical reference called BIRD, stands for Biologically Interesting Molecule Reference Dictionary. On the right side are two examples of the BIRDs of selstreptin and gramicidin. So these molecules are usually short oligomers of peptide inhibitors, antibiotics, or well-known carbohydrates. And they may have chemical modifications on the components or on the bond. So it's kind of non-standard from regular protein or nucleic acid. From different perspectives, data users may refer them to as a short sequence or as a single molecule description. Therefore, WPB provide dual representation in the BIRD reference so that user can search either way. And we use this BIRD reference to do our OneDep annotation and deposition service so that the same compound will be represented consistent across the PDB archive. There's another type of oligomers, that is oligosaccharide. Oligosaccharide is different from peptide and nucleotide because they can form branches for 1s to 1,3, whatever. So that the sequence alone is not adequate to describe their chemical identity. And it was challenging, but we managed to consult the glycoscience community to adopt a modernized scientific representation of these glycans as branched short oligomer with monosaccharide sequences. Of course, each monosaccharide is defined in CCD. And we also provide community supported linear descriptors. On the right side, an example of N-glycan core on the SARS-CoV-2 spike glycoprotein. The different shape and colors are ISNFG symbols for different monosaccharides. And on the top, you see the 3 formats of linear descriptors adopted by the glycoscience community, and they are machine readable. We are now reviewing the metallic ligands in the PDB. I mean, there are 63 metal elements in the PDB. Some of them are single metal ions, like hydrogen, whatever. But beyond that, we have complex metallic ligands, such as the one on the right, and hundreds of PDB entries with them. So we're working to review this and make sure they're represented in a consistent way. In annotating the small molecule ligand data, we follow the FIRE principle as stressed by Stephen and Ezra. So we want the data to be findable, accessible, interoperable, and reusable. Therefore, we try our best to provide accurate chemical description in the chemical component dictionary with standardized format. And we ensure consistent chemical annotation and representation across the entire PDB. And we go further to review the experimental data support for the ligand structures and the interaction between the ligand and targets. And finally, we also develop softwares to help users to search and analyze this ligand data. In terms of bioaccuracy steps, our goal is to provide accurate chemical description and make sure that outcome data follows the FIRE principle. So they also provide small molecule structures are analyzed and searched against the CCD. We will have 2D or 3D comparison view, which we will see in the demo. And to provide comparison for the author provided and CCD matches, and review the chemical descriptor provided by authors, such as drawings, names, etc. We review whether or not the chemical identity is correct or possible. So if we find a match in a CCD, we standardize first by assigning the CCD ligand identifier to that specific unique compound. Or if the compound is new to the PDB, we create a new CCD definition, and to make sure that the future deposition will follow the same annotation. We check data quality by examining the goodness of fit between the model and experimental data. And we also review the interaction between the ligand and protein to create a link record if the ligand is covalent bound to the protein. And if the compound is kind of like short oligomer, we also create a BIRD reference for due representation. Now, when users come to review the ligand structures in the PDB, it's important to know there's some features in the PDB ligand structure that is unique, different from other databases. For example, a ligand can be either non-covalent bond or covalent bond to the protein. Here on the left side is a penicillin G in its unreactive form bound to a mutant from this penicillin isolase. On the right side, it's also penicillin G, but it's reactive. It's covalent bond to the beta lactamase. And from here, you see the beta lactam ring opens up and form a covalent bond with the serine at the active site. Now, in the PDB model file, we explicitly identify this bond and record its covalent connection, so users can search for it. Even though a ligand may bond to different targets, both non-covalently, but they may adopt different conformation. In this example, the coenzyme NAD is bound to one target in the fully extended form on the left side, and the right side with a different target, you see it's wrapped around and make a more compact conformation by forming this pi-pi stacking. Now, even though the ligands in different structure may adopt the same conformation, they may have different environments. For example, this is the ligand TDT that is a peptide-like protease inhibitor. But when you compare the ligand binding pocket in different species of the coronaviruses. In this case, the purple with SARS, the blue with SARS-CoV-2, and the cyan with MERS. You see that the binding pocket had different orientations on the residues, thus impact interaction between the ligand and the protein. Those are other features I want to draw your attention. For example, about 6% of ligand in the PDB are kind of incomplete. Some atoms were not built because the experimental support was lacking on those parts. And also, when you review a ligand, it's important to review the ligand structure quality. The overall structure quality indicators, such as R and R3, may not be adequate to indicate the quality of the ligand structure because they are the overall indicator, and ligand is a local structure. So there's local indicators such as RSC and RCC for small molecule ligand structure. And you can even go further by manually review the goodness fit between the model and the experimental data, which I will show you in the demo, in the tutorial. You can download the entire CCD reference from WPDB either through HTTP or FTP linkages. As you can see on the right side is a screenshot where you found it, the CCD and the bird, and same with the bird and a different format. So you can also visit each individual definition of the small molecules at either our FTP or individual partners' website from RCSB, PDB, and PDBJ. On the right side is an example of RCSB on ligand PN conditioning G. Looking forward, first, as I said, I want to provide uniform representation of metallic ligands. But more important, as Ezra has raised, we will extend the CCD identifier beyond 3-letter code because we just use up the combinations. And PDB format is limited to the 3-letter code. Therefore, the PDB format will be invalid when we go beyond 3. So we encourage users to remove such limitation and start using mmCIF format for their researchers. And that concludes my talk. Thank you.
Thank you, Chenghua. I don't see any open questions in the chat. So at this point, let's take a 7-minute break and reconvene at 20 minutes past the hour. That would be 20 past 11 Eastern Standard Time and 20 past 4 UK time. And we will then go through the tutorials with you. Thank you very much for to the speakers. And I'm just going to share a slide briefly to acknowledge. Can you stop screen sharing, please, Chenghua? Thank you. And I will just briefly acknowledge the other members of the RCSB Protein Data Bank who made these presentations and the tutorial possible. Thank our funders, the National Science Foundation, NIH, various NIH institutes in the United States Department of Energy, our hosts at Rutgers, UC San Diego, and UCSF, and of course acknowledge the important role that the Worldwide Protein Data Bank plays in everything that we do. So we'll be back at 20 minutes past the hour. Thank you.
Bailey, do you want to just say a few words?
Yeah, can everyone hear me okay?
Yep.
Perfect. All right, so thanks to Stephen, Ezra, and Chenghua for the excellent workshop that we've had so far. Just to introduce myself, my name is Bailey and I work for Liverpool ChiroChem, and we are a chemical technology innovator on a mission to accelerate small molecule drug discovery through expanding access to 3D chemical space. And at Liverpool ChiroChem, we are proud to sponsor this series of workshops as we understand how essential that these open source tools are for the global research community. At LCC, we specialize in using stereo-defined N-heterocycles for use in DEL, FBLD, bifunctional linkers, and our 3 Discovery virtual library. If you would like to find out more about how LCC could support any of your projects, then please reach out to us through our website, liverpoolchirochem.com, or you can find us on LinkedIn as well. I hope you all enjoy the rest of the workshop, and thank you for your time.
Okay, thanks.
Okay, it's now 20 minutes past the hour. Let's resume. I hope everybody's had a chance to have a quick bathroom break or get another cup of tea. So Ezra will be beginning the tutorial with some tips on how to use OneDep, and then Chenghua will follow him, and we'll conclude at the top of the hour, to be respectful of everybody's time and other commitments. Over to you.
Okay, thank you, Stephen. Everyone hear me?
You're good to go.
Okay, good. So Chenghua and I are going to discuss two quick little tutorials on the OneDep deposition in small molecules, searching within various resources. For mine, we do not have a sample server that you could just play with, but you could use our validation server for some of this process to see what type of things would come out with reports. So the first thing it comes about is you want to start a deposition. You sign into our deposit system, deposit.www.pdb.org, and you are asked first for what country you're in. This is our geographical redirection page. I'm not showing you a picture of it right now, but then it would put you to one of our 3 primary deposition sites, either in the United States, UK, or Japan. You will then be asked to give information, like who you are in terms of contact. You can come up with a password. This is a shared password that's used by all depositors within your session. So if you have collaborators who are going to be accessing this, don't use your Gmail password. Don't use your bank ID number or anything else like that. If you leave it blank, we'll issue a totally random one. You tell us what type of experiments you want to do. This essentially gives you what type of IDs. If you're submitting, for instance, electron microscopy, you'll be getting an EMDB ID. If you have a model associated with it, you get a PDB ID. If you have NMR, you'll also get a BMRB ID when you're done. You check off on the terms and conditions. We have some privacy policies, just so you acknowledge that you've received them, including what data we will or will not share with people. You then will be sent, once you press start deposition, you'll be sent an email message with, here are your login credentials. You enter them in here, and you'll be able to then get access to the system. Within the system, we have what's called a left-hand navigation bar. Initially, it looks very sparse. We have overall instructions, a communication facility within the system that you can send messages to the biocurators while you're depositing your entry or afterwards to ask questions if you have any difficulties. But the first thing you'll want to do is upload your files. You could copy information from a prior deposition you made if you have a previous deposition ID, such as your contact information, citation information, or grant information, because you don't want to type that in again if you don't have to. You'll then proceed down to the file upload section, where you will upload individual files. You can do simultaneously, and then you will have the ability, you have to specify what are these files. We are not going to guess, oh, this looks like a PDB file. This looks like an mmCIF. You have to tell us what you're uploading. You then tell us to proceed, and we will then do some various checks, convert the files, make sure that it adheres to the proper format, and then we will start a structured validation. Once that starts up, This takes a minute or two. You then come back in and you will see a much more enriched left-hand menu with green check marks, meaning, okay, you're good there, or red areas, which means we have missing data, mandatory data that you have to fill in. How much mandatory data? Well, across the top is sort of a description of, well, it looks like we're here, we're about 50% done. While you could potentially have, much more data, we don't require all of it to be collected. A bunch of it is optional. So if you look at, for instance, here, which is the information on reflection data, you may not have from your data processing software CC1/2 or CC star. That's okay, it's not mandatory. But we do require, because it's mentioned in red, the unique number of reflections that were measured for an X-ray experiment. And if you don't put in a value, we will block you. We also have the ability to provide advisory messages. So an R merge on I know is 0.4 is very unusual, but there may be something unique about your structure that we don't know about your data collection. So we don't want to block people, but we want to highlight it that, hey, this looks unusual, and maybe you want to double check that you didn't mistype a number. Maybe it was 0.14, not 0.4. We don't know. It also ensures that you don't wind up with some people who used to put in percentages. So if you had 4, is that 4% or 0.4? We don't know. You eventually go through all the metadata, try filling in all the data you need, including how you want us to hold on to it, your titles, your citation information, macromolecule information, data collection statistics, where you collected your data, was it a home source, a synchrotron, and various other metadata that we can incorporate into the final file. The goal is that we do not want to have to make these changes. We're allowing you to put in the correct numbers, so you don't have to later tell us, oh, I made a typo over there. You're seeing it all while this is the data that will be incorporated into the final file. So one of the tasks that you're going to need to do is, if you have a ligand or a small molecule that we have not seen before, or even if we've seen, you have to make sure that all of them are accounted for. In the older prior systems, you would just come through your deposition and we would try to figure out behind the scenes later on, okay, what does this ligand really look like? We extracted the coordinates from the chemical component from your coordinates. What's the bond order? I don't know. We do our best, but right now you're given an opportunity to give us much more detailed information, so we'll get it right to start with and not have to go looping around between the depositor and us. You will have the ability to indicate, so you start off here where you have a summary of what ligands there are. You'll have a chance to say, what is our ligand of interest? What is the purpose of this investigation? Was it just something that's floating around? Zinc. You have a zinc finger protein. Well, that's structural. That may not be important to what you're interested in. You're interested in binding a small molecule that disrupts a DNA, which is attached to, which is being held on in place by a zinc finger. So therefore, you're probably not going to say zinc, but whatever ligand is, you would say might be my ligand of interest. Now you're going to have 3 choices of how to resolve these issues. Either it's a match, so something that's simple, arguments, let's say ATP, you probably will not have to do anything with. It'll be fine, and it's like the chloride ions here. You might have to address the match by saying, oh, it's really another element that we have. I'll show you that example in a moment. Or maybe you're going to have to give it a more detailed description. So here's an example of our ligand TTTT. It came in as lig, but really should be, but it says it might be TTT. We'll present you with a 2D representation of what we extracted from your model coordinates, what we think it might be, and you can see it's 2D or 3D. If it's a proper match, you tell us, and then you just say that, okay, we're done with this instance, and we're done with all of the ligands if it's relevant, or you go on to the next one. If we don't have any samples of publicly available ligand definitions, you could tell us information about it. We could tell us, is it peptide-like, is it carbohydrate, is it lipid heterogen? Or you could give us an alternate ligand ID if it's present in there. Or we would request that you give us information like a picture or a smile string that we could then know what were you working with. And this is much more reliable for us on the back end. You'll eventually get down to validation reports. We require that you download a PDF report that we've presented. We highlight on the deposition system some of the major issues that were found. Like here, there are a few residues which have some unusual electron density. You may want to look at it. In the end, on the bottom of the page, you will be able to check off that you've reviewed it. During deposition, you'll get a preliminary validation report, and that's not to be submitted to the journals. We'll give you a confidential one during the actual review after we process the data and send it back to you. In the end, the last thing to do would be signing off on the terms and conditions. Essentially, it's a lot of text, but essentially it's you and your collaborators are all aware that what's going on, pretty much. You're all co-authors. The PI is aware of it and in charge of everything. You've looked at the validation reports. You understand our policies, and you understand that in something new this year is that the principal investigator's contact information will be made public on release of the entry. We're talking about name, email, and ORCID ID. So if there's a question, people know who to write to ask questions about this entry. We'll then send data back via the communication tab. This validation report, you could ask them, say, hey, I need to replace my coordinates. Perfectly fine. About 30% of depositions do this. If everything is fine, you write a communication that says, okay, we're all set. There's a task for doing that. And then we pretty much put the structure on hold until it's ready for release. And there could be several reasons. You have an expiration deadline, hold it for 6 months, or it could be, we found the literature citation was published online. Another user tells us about it, or we receive a request from you or the journal to release the information, the model once you're done. And that's all I have to say right here. And I'll now turn it over to Chenghua so he can move on with how to explore ligands. This is more of a hands-on tutorial.
Thank you, Ezra. I don't see any questions in the Q&A, so let's continue, Chenghua.
Chenghua, I think you might be muted.
Chenghua, you are on mute.
Okay, sorry about that. Thank you, Ezra. So from this part, I will talk about how to find and explore a ligand in PDB. So I'll start with how to search a ligand. We can search the ligand either by CCD identifiers, their names, descriptors, type in. We can do chemical drawing search. And for the search, we can choose different match options. For example, the exact match, similarity match, or substructure match. We can also display results in different ways that we wish. And then we can explore the ligand by review the CCD ligand definition, the chemical information. And some of them have external resources from other databases such as drug bank. And we can review the interaction between the ligand and the protein. And finally, I will provide 2 short exercises for both of them. Now, first, I want to stress that the exploration of ligand in the WPDB is offered on all 3 partner websites for search and visualization. And as you can see here, in terms of ligand interaction, all 3 sets, RCSB, PDB, and PDBj provide their unique features from different perspectives. And in the following, I'll focus, I'll use examples at RCSB, but you can find similar functionality on all 3 sets. Now, at RCSB, there are 3 ways to search chemical data. The most general search, the text search from the top bar. The advanced search, where you can either search the chemical attributes, such as names, synonyms. You can also search the chemical descriptors here. And you can also use a chemical sketch to draw or modify the ligand, then search. We start with top bar search. Now, you can, from here, you can search ligand ID, chemical names, synonyms, etc. So on the right side of the example, we just want to type penicillin as a keyword search. And then you click search, and you will find structures that has the penicillin somewhere in their text. But you can display results by focusing on the non-polymer entities with this keyword. And then you see that 1G-M7 show up as the first hit. And this one has penicillin G as the entity number 5. It's number 5 because there are two protein polymers. There are also other non-polymer components. So the penicillin G is the number 5. And you can see the summary here that the chemical ID is PNN, which I showed in my small molecule talk, and their formula. If you go to advanced search, you can do more focused ones. You can specify what you want to search as a chemical name and synonyms. In this case, example, I searched the same penicillin, but I want to focus on the name. Now, from here, you have results, and you can display the results in various ways. For example, you can display the results as molecular definition, which returns to you the CCD components, the CCD small molecule definition that has penicillin in the name. You can also display result as the structure as non-polymer entity, which returns to the structure, PDB structures with these ligands, as you can see from here. And just similar to the last page, it shows the entry with entity 3 and penicillin G. You can also search chemical descriptors. From here, you can search like either smiles or inchi. And now you have search options because you're providing explicit description. So you can do exact match, you can do subtraction match, you can also do similarity match. On this example, we search this smiles, and want to do the similarity match. And when you do it, you get 7 returns, 7 hits. That have the chemical structure similar to what's described in these smile strings. Now you can also do chemical type matches from enumerated list. And you can display results differently, either a gallery view or summary view. In this example, I want to find all the disaccharides with beta anomeric linkage. So here you return all these monosaccharides, that is beta D monosaccharide. And if you want to search all the peptide-like small molecules, you use the type peptide-like, and here it's the return displayed as summary, which you see not only the drawing, but also some short description of these molecules with formula, inchi key, etc. Finally, we can do the chemical drawing. So the site provides a 2D panel. I mean, for you to draw the chemical in the 2D scheme. And you can also shorten this by loading some descriptors. In this case, you can load either inchi smiles and display them as drawing. Then you can modify it, which I'll show you in the tutorial. And again, you can choose different matches, either similar ligand, substructure match, exact match. They are under different algorithm. And more importantly, when you see the return to results, it's actually a query that is already formulated. And you can combine this query with other searches. For example, I want to find this ligand with a protein named ABCD. You can actually add that search as a combined query of protein ABCD together with ligand search and find exactly what you want. Once you find the ligand you want to explore the ligand. And first thing you want to see how this ligand is defined. And as we know from the talk that ligand is defined in a CCD in mmCIF format, and it provides rich data content. So you can search this ID from either top bar, when you tap top bar, and in the drop-down menu, you can narrate the chemical ID search, reach the summary. And you can also go on a structure that has this ligand and scroll down to the small molecule section, and you click on the small molecules identifiers, it will also bring you to the ligand summary page. And on the page, what you see are the data contents in the CCD, in their CCD definition, plus the 2D and 3D view. In this example, you see for the TTT PL-Pro inhibitor, you see the name, identifier, formula, molecular weight, SMILE inch, etc. And beyond that, you can see the third party, external resources related to this ligand. For example, TTT actually is an experimental drug that has drug bank ID, this link out to drug bank, and have drug target, drug targets. And you can also find other information for this experimental drug. You can also explore the information from, as long as they are applicable, from CCTC, Campbell, CABI, binding DB, etc. One of the most interesting things about PDB structure is how the ligand interacts with macromolecules. And the most straightforward review for this is the pocket view. And here we provide a one button click, you can reach this view. On the structure summary page, if you scroll down to the small molecule section, you have this ligand interaction button. Once you click, you reach the interaction view that shows focus on the ligand, zoom in, and shows the interaction between this ligand and the surrounding environment. And you also have the option to show the electron density to see how good the experimental data supports this ligand. Again, I want to stress that all three websites from RCSB, PDB, and PBA have similar features, kind of from a little bit different perspective. And you can explore the ligands, search the ligands on all three sets. And if there's no question, I'm going to the exercise. Do I have a question?
There's nothing in the Q&A.
Sure.
Please
Okay.
proceed, Chenghua.
So for the exercises, if you can, I would like you to open a browser on your machine, and I'm going to do the same. So here are the steps. So what you need is, okay, Jasmine already copied it there. Thank you, Jasmine. I'm going to exit Zoom and do the exercises here. Here's RCSB.org, where we want to show how to find structure with chemical drawing. So you go to search. From the search dropdown, there's this, as I said, they're basic, advanced, and there's chemical sketch search. Click this, and it brings you to the chemical sketch tool. Now here, you can draw the ligand you want to search. To make it short, I will just load a descriptor. And this is SMILE descriptor. You can also load the engine. So now from here, you have different ways to modify the ligand. Let's say I just want simple addition and add a methyl group here, opposite to the amino. Now, assuming this is the ligand you want to search, and you want to search this ligand in the PDBC chemical component dictionary, scroll down, you have this search option. You can either search by INQ or SMILE, and you can use different match options. I'll just use default. And from here, you find 4 ligands that are similar to the drawing I just provided. And now let's say I just want exact match. So you change the match option to exact match. And you click search again. It finds the exact one you want. This is the DDT. I've been consistently using as piping-like protease inhibitor in SARS-CoV-2. So now you simply click this code. And it brings you to the ligand summary page that I will talk about. On this page, you see the 2D, 3D view that you can do different review. And you see that it displayed the CCD annotation data, the name, smells, INCHI. And you see also external resources from drug bank. If you click here, it will bring you to drug bank and PubChem Campbell. And this concludes the first exercise. If you have any questions, feel free to ask. You can either post in the QA. Do we have any questions? We'll go back to the journal. Do you have any questions?
There are no open questions.
No question. Okay. Then we go to the second exercise, which is how to explore ligand interaction on the pocket of the ligand. So let's say, assume you find the ligand PDT, or you find the structure. You can find the structures with ligand PDT. Now, on the ligand summary page, you have this link of how many entries have this ligand. And here you find the file structures. 4 of them are SARS-CoV-2. Let's say also, I mean, you can find the structure of interest, for example, this SARS-CoV-C11S mutant with the inhibitor. You may also read the paper and you just see the PDB ID and you want to type it to IR. And you do PDB ID search, you reach that structure summary page of the 7J IR. This is called, this is a mutant with the peptide protease inhibitor. So if you scroll down to the bottom, you first see the macromolecule description, then you see the small molecule description. And here is the ligand interaction button I would refer to, and click that. Now, for here, it brings you to the 3D view in MOSTAR. And from here, you can see the ligand is focused, ligand PDB, and all the interactions of hydrogen bonds, pi stacking, etc., separately marked. And if you want to explore how good, so this model is supported by experimental data. You want to, in this case, is actually there. So you want to show the electron density view. So you go to MOSTAR density view, click enable on the right side, and then you show the density. Now we zoom in again on this ligand. So now you see the view of electron densities. They're different maps. And I want to just remove the differential map, focus on the F4 FC map. And you can actually show it with warfarin, most people see. Now you can see that this ligand is well supported by electron density. So its conformation is fairly accurate and confirmed by the experimental data. And that concludes my second exercise. Thank you. If you have any questions, feel free to ask.
Sometimes you get multiple conformations of ligands. How do they appear annotated?
Multiple conformation in terms of ligand, if they build with like alternate conformation, like AB or ABC, it will show up all of them combined. You will see the alternate conformations, and then you can see the density to see whether or not the alternate conformation is supported by electron density. Usually you see a kind of blurred electron density for 2 different partially overlapped conformations.
So during annotation, there is an alternative alternation identifier that we can label for this case.
You can also view a specific conformation. In this case, I mean, there's one conformation for simplicity of region. But if you scroll down on the bottom right, you actually see the conformation if there are multiple.
Okay.
And your questions, right?
So the question is, can we see an interaction with the metallic complexes with proteins with 3D representation?
So you want to see example, right?
Yeah.
Okay, you just do that. So this is our Shao cluster, the common one, the most common one, the cubic, pretty. So you can find structure with all of them by just clicking them. And I just randomly try to find an example. We try to find let's find the most recent one. And the thing with electron microscope has become more popular. So the top list usually have a lot of, the most recently have a lot of electron density, electron microscopic structure. So we will choose this actually at 1.5 angstrom. And same, you just, from the structure summary, I'll just maximize this. From structure summary, you go to the small molecule section. And you just click this one button. Here, you can see the cluster interacting with the environment. In this case, you see kind of replacement software from the cysteine, I think. This is cysteine. Not replacement, the interacting software. And it's obviously not perfectly cubic anymore. But you see the interaction between this Airsoft cluster and the protein target. I mean, you cannot say target is the macromolecules is associated with. Does this answer the question?
I think so, yes.
Okay. Anything else? Nope. So shall I stop sharing?
Before you stop sharing, Chenghua, I suggest that you go to 1 alpha alpha Yankee. That's a zinc finger DNA complex from Carl Paber's lab at 1.6 angstrom resolution, published in 1996.
Okay.
And it would be interesting to see the environment of the zinc there with the electron density.
Yeah. This is zinc. So
yeah.
Now you want to see the electron density, right?
Yeah. So this is these zinc fingers can coordinate the zinc with 2 cysteines and 2 histidines. So let's see the electron density. Oh, there were no structure factors. Of course, the structure is too old.
You want a different?
I'm sorry. I'm sorry. Let's try 1 Juliet Kilo 1.
So let me see if it's
same it's a mutant of that same protein.
But this one doesn't have how accurate data as well.
Okay, there all the structures are too old. Yes.
So you want zinc finger?
Well, I was thinking about a zinc finger, but
search for zinc finger in my name.
Does anybody in the audience have a favorite zinc finger protein, a modern one that they would like this demonstrated with? So what about 2-BKF? Does that have structure factors associated with it?
2
B Bravo Kilo Foxtrot.
This?
Yeah.
No.
No.
1P, go for 1P47. He has 1 Paul 47.
Yeah, okay, good. So we want to see the zinc. Okay, now we see the density. Exciting. That's it.
Yep, very good. Thank you.
It's prettier.
Yeah. So this little exercise underscores the fact that in the early days, even though deposition of atomic coordinates became mandatory, deposition of experimental data was not. But in the modern era, deposition of the atomic coordinates and the experimental data are a condition for publication in most scientific journals. And we found that our depositors, the many tens of thousands of them, seem to be very happy to comply with these mandates and provide complete information for the structures that they're depositing to the PDB.
Yeah, Ezra said this is Ezra's structure. He deposited, so he followed protocol.
Okay, so that's how we know that it was done properly. So at this point, if there are no further questions, either in the chat or in the Q&A.
Could I ask a quick question?
Yeah, please do. Please go ahead.
So, as you said, a lot of these are very old structures now. Is there a process for perhaps, or what is the process for correcting errors in older structures?
So we do have a mechanism for the depositor of record, the principal investigator of a particular study, to deposit a new set of atomic coordinates if they seek to make a correction. This is called coordinate versioning, and it was introduced a few years ago. And we've been very gratified by the number of taken the trouble to go back and correct the scientific record when they realized that there was an aspect of the original structure determination that may have been suboptimal. In many cases, it's because of improved technology that they're able to use the same set of structure factors and do a better job. The archive is growing, is doubling every 7 years or so. So in fact, the number of very old structures with incomplete data is a small proportion of the entire archive. And every year, that proportion becomes smaller and smaller. In many cases, either the same structure or the structure of a homologue has been done recently. And so the PDB user may wish to order the results of their structure search in terms of going from newest to oldest, and so they can be sure to pluck one of the newer structures. It's also the case, and this was documented in one of Chenghua's papers, that the average quality of PDB structures has been slowly improving over the years, particularly for the atomic coordinates of the macromolecule. more attention is now being provided to, more details now being provided for looking at co-crystal structures with the goal of trying to raise awareness about the importance of proper refinement of the small molecule within the 3D structure. And we're seeing improvements in not only in the atomic coordinates for the polymer components of structures, but also for the small molecule components. And I think we are at time. So if you could just stop sharing, Chenghua, please.
So before I stop, just very quick, 1 second, to add to Steven's information about the updating of the structure. If there is PDB not only try to make sure the data is accurate, but also will record the history of any data set. In this case, the 1P47, the one I just showed, there is revision history. And if an author wants to update the correct errors and change the coordinate or something, it will also up here. So, and there are more information on this.
Right. Okay. So please stop sharing, and I will just reshare our acknowledgement slide and thank, in particular, our PDBE partners for working together with us and the Royal Society of Chemistry to put on this 2-day event. And the PDBE team will be taking the lead on Thursday morning, same bat time, same bat channel. So thank you very much to Ezra, to Chenghua, to the Royal Society of Chemistry, and to our corporate sponsor for making all this possible today. And thank you to the participants for showing up, and any feedback you want to provide in terms of the value that you received from this event can be sent to our customer service desk, which is emails here, info@rcsb.org. And all feedback is good. Positive feedback is nice to receive, but constructive criticism is even more welcome. Thank you to one and all for making this a great success.
Yeah. Well, thank you very much. I think this has been another fantastic session. I'm a regular user of the PDB, so I'm one of the things that always strikes me is that every time I hear one of these sort of tutorials and things like this, there's something new I learn about it. It's there's a huge amount of data and a huge amount of information in there, and getting access to it all in a free and open manner is absolutely fabulous. So once again, I reiterate my thanks to all of the people who presented today. I think it's been a great day, and I look forward to hearing more.
Yep. See you back here on Thursday.
Right. See you then. Bye.
Bye.
Bye. Thank you.
Bye.